Thank you, Maurice. It's a great honor to be here to give the Maxon Lecture. I had the opportunity to meet Professor Maxon this morning. He's a wonderful gentleman, but a bit too modest, because only just now did I realize about all of his wonderful accomplishments. And I'd like to thank my hosts, both from mathematics and computer science. At the dawn of the Human Genome Project in the early 90s, I decided to turn my attention to computational problems in molecular biology, thinking that my background in algorithms and mathematics would be useful. It's turned out to be not as easy as I'd thought, but I want to tell you a little bit about this field, including some of my own work, but also the work of others. And I'll begin by saying that changes have the manner in which biological research is done, particularly in molecular biology, has been revolutionized by a number of factors over the last few decades. And the goal is to derive a much more detailed quantitative description of biological systems and understand the molecular processes of life, the applications to medicine. And these developments involve a number of fields, as I have written here: biological, physical, engineering, and mathematical sciences. For those of you like me who never took high school biology, let me give you describe this cartoon of a eukaryotic cell. The term eukaryotic refers to cells in which there is a membrane separating the nucleus of the cell from the exterior, what's called the cytoplasm. The DNA, which codes for the proteins that do most of the work of the cell, resides in the nucleus. Every cell in the body has the full complement of DNA, the so-called genome of, in humans, 3 billion symbols called nucleotides, A, C, T, and G, the basic components of this big macromolecule. The DNA doesn't really do anything. It's a passive repository which contains the description of the proteins that the cell makes and a description of the mechanisms that regulate those proteins. The conversion from DNA to protein involves two principal stages: a process called transcription, in which the parts of the DNA, parts of the genome, are selectively chosen and transcribed in a letter-by-letter fashion into the four-letter alphabet of RNA, a different molecule. DNA is a double-stranded molecule. RNA is single-stranded. The RNA is transported outside the nucleus and at a molecular machine, or molecular machines called ribosomes. The RNA is translated into protein, whereas the RNA can be thought of symbolically as a sequence of nucleotides of 4 types. A protein is a sequence of symbols which are of 20 different types. And so a protein at one level is a long chain of symbols from a 20-symbol alphabet, but the functioning of proteins requires a description of the three-dimensional structure into which the protein folds. And so the story I'm going to be telling will deal largely with the interactions among these molecules. The process of transcription will be described a little further, and a little bit about how the proteins in a cell regulate its activity. So here are some of the goals of the study of the genome and the associated molecules. We want to sequence and compare the genomes of many organisms. A great landmark around the year 2000 was the sequencing of the human genome, which was supposed to open the door to all kinds of wonderful applications, but some of those still are under development because reading off the 3 billion nucleotides by itself until you interpret their semantics doesn't tell you very much. And so we're still understanding what that long sequence means, identifying the genes within it, determining the functions of the proteins they encode, describing the process of going from genes to proteins, and understanding how these macromolecules work together to execute algorithms which control the fundamental processes of a cell. Other goals are to trace the evolutionary history of different organisms, to see how we evolved from earlier organisms which may not survive on Earth, to understand the function, structure, and evolutionary history of proteins, and to study the associations between genetic mutations and disease. So among the structures that we try to analyze in computational biology, there are the phylogenetic trees, which describe how the various kingdoms of life emerged and the differences among the different species that have emerged in the course of evolution. And so a fundamental computational problem is to look at the leaves of such a tree, which I'll refer to as species, although here they're actually bigger domains of life, to look at information within those individual species, and then to figure out from that the most likely manner in which those species eventually developed by a process of branching off from ancestral species. So there's a whole computational area related to the inference of these phylogenetic trees from typically DNA sequence or RNA sequence information in the extant species. Fundamental to the whole enterprise is extracting the sequence of a genome, and there has been great progress in that area. Basically, all of these different methods of sequencing a genome start by shattering many copies of the genome into smaller pieces, reading off those smaller pieces, and then combinatorially figuring out how the pieces line up to comprise the overall genome. We've progressed over the last couple of decades from sequencing small bacterial genomes of size a few million nucleotides to fruit fly, human, mouse, mosquito, rice, and many others. It turned out that a technique called whole genome shotgun sequencing became the best computational method. And recently there have been dramatic advances in sequencing technology, which brings within sight the prospect of actually reading off an individual's genome at the cost of $1,000 or so. We haven't quite reached that point, but when that happens, we can predict that your medical record will consist of your entire genome, and that information about the differences between genomes of individuals will influence the way they are treated medically. In order to understand what's going on in living cells, in addition to having the genome, we have to understand which pieces of the genome are being transcribed into messenger RNA, and a technology called the DNA microarray allows one to measure the abundances of many tens of thousands of different species of RNA to see how much of those different RNAs are being manufactured by the cell, which gives a sense of how the cell is behaving. And then we have to measure how the various molecules interact to form molecular machines. So we look at protein-protein interactions, protein-DNA interactions, protein abundances, and protein structures. And we can also be guided by imaging techniques which allow us to actually look at a developing embryo. So here's a zygote, the originating cell which develops when a sperm and an egg meet. And I show this only to indicate the detailed kinds of imaging that one can obtain, images that one can obtain for these individual cells. So you can look, for example, at the whole body plan of a worm and see all of the parts by means of imaging. The field also develops through advances in databases so that scientists don't have to do everything from scratch, but can look at the data that's been gathered, genomic sequences, protein sequences, algorithmic tools for searching through a database of sequences to find those sequences that are most similar to a given sequence in order to understand what that given sequence may be doing. Finding the genes within a genome, and studying, as I said, the interactions between the macromolecules and so forth. Various kinds of databases are being compiled. And finally, advances in mathematical modeling. So we're now in the era of probabilistic machine learning, which has become perhaps the dominant sub-area of artificial intelligence, and in particular, probabilistic models and machine learning techniques are very useful in exploratory data analysis, pattern recognition, and so forth. And we also, as I will describe, need algorithms for manipulating phylogenetic trees, sequences, networks of proteins, and other combinatorial objects that arise as we try to understand the systems that these molecules form as they interact as molecular machines. So as a warm-up, let me talk about one of the earliest and most influential algorithms in computational biology. This is the algorithm for computing what's called the edit distance between two sequences. So a recurring theme in the field is that you would like to measure how close, how similar two sequences are. So, for example, if you have a sequence whose function is known in human, and you want, or say in mouse, and you want to understand whether there's a comparable sequence in the human genome, you have the problem of measuring the dissimilarity between two sequences. That's a recurring problem. And we can define the dissimilarity or the so-called edit distance as the number of insertions, deletions, and substitutions of symbols needed to pass from one sequence to another. So in the example, if we look at how many of those primitive moves are needed to take you from the sequence AGCAT to the second sequence written here, you can represent the changes by what's called an alignment, where you insert space symbols to align in one or another or both of the sequences in order to exhibit their similarities. And these spaces can be interpreted as deletions. So if you're going from the first sequence to the other and you look at an alignment, we see that they share the symbol A in the first position. Then you have in the next two positions of the alignment space symbols. So those represent the deletion of the C and the T in the second sequence. Then you have the G's agreeing, the C's agreeing. And then in the next to last column, you have an A in one and the T in the other. So that can be interpreted as a substitution in going from the first sequence to the second. And then you have agreement in the last position. So we see that there, in this particular case, in going from the first sequence to the second, there are two insertions and one substitution. And that turns out to be the shortest way to do it. And so the edit distance is 3. And there's a quadratic time dynamic programming algorithm to compute this edit distance between sequences. So the running time for it is basically the product of the lengths of the two sequences. And it uses the fundamental technique of dynamic programming. The basic idea underlying dynamic programming is to solve a big problem. You solve a similar problem on a large number of suitably chosen subproblems in such a way that you can move through the larger and larger subproblems to compute optimal solutions to all of them at minimum cost. So in this particular case, to compute the edit distance between two strings x and y, you make a table with an entry for every pair consisting of a prefix x prime of x and a prefix y prime of y. And then using a simple recursive formula, you can just go through and in constant time for every entry in the table, you can compute the distance between the two given prefixes in terms of the preceding results for shorter sequences.
What's a prefix?
A prefix is just an initial part. So a prefix of the word borderline is the word border. Yes, just an initial part of the word. So you have to go through all pairs consisting of the prefixes from the two strings. I won't elaborate on the details of the algorithm, but computing the edit distance between two strings, or equivalently computing this optimal alignment, is essentially an easy problem. On the other hand, we could be interested in the common alignment of many sequences. So there, what you are again trying to do, here you have the sequences of many different related molecules. Each row of this matrix of symbols is the underlying sequence, in this case RNA, for one of these molecules. And you see here and there spaces within it, space symbols. And the alignment, again, is according to a certain objective function that I won't define, attempting to get the least costly alignment of all of these sequences, the alignment that best displays their similarity. So the perfect alignment would have columns where all of the symbols are the same, but you pay penalties whenever you have spaces or whenever you have symbols differing in the same column. And it turns out that the problem of computing an optimal multiple alignment for a set of sequences is NP-complete. And in fact, most of the problems that arise in computational biology, computational molecular biology, are NP-complete. And what this means is that we cannot expect to get highly efficient algorithms for solving these problems optimally. And for that reason, in computing phylogenetic trees or many of the other, or multiple alignments or many of the other problems that come up, we have to use heuristic algorithms. And one of the great mysteries in the field of algorithms is to put the study of heuristic algorithms on a sound scientific footing. One of the most important programs in computational molecular biology is the BLAST program, which is related to edit distance, but a little bit different. Instead of just having two sequences, you're looking at one sequence versus a whole database of sequences. And BLAST uses clever techniques from computer science to search the whole database to find those sequences that have an exceptional similarity to a given sequence. So BLAST is a workhorse that molecular biologists use. I already referred to the process of, I hope you won't mind if I do a Marco Rubio imitation for those of you who saw his speech at, after the election. He was criticized for sipping water in front of a camera, but there, I've done it. So sequence assembly is the process of figuring out a very long DNA sequence from randomly generated short sequences called reads. Some of these reads come individually from the experiment, from the basic experiment, which involves some very elaborate, highly parallel optical techniques. Some of these reads are individual, and some of these reads are paired, which means that you read these sequences and you also find out from the experiment what the approximate distance between them is in the genome. So the process of sequence assembly is a little bit like the following. Imagine you have many copies of a book, and the pages are torn out of these copies and they're shredded, and then they're rained on a little bit, so some of the letters and words become indistinct. And then you have the problem of looking at all of these soiled copies of the book and trying to figure out what the text of the book is. That's basically what the problem is. But it's exacerbated in the case of genomes because the DNA book has many repeated sequences that occur over and over in the DNA. And those repeats mean that whenever you can no longer assume when two reads overlap that they come from the same part of the genome. They may come from different repetitions of the same sequence. So it's a very difficult problem. The final objective is to take these millions of reads and to compute the sequence of the target genome. The difficulty is that the reads are noisy. The pages get rained on, so to speak. You have repeats. And there is a complex strategy that has been evolved called whole genome shotgun sequencing. A pivotal change in the landscape of the field came when it was discovered that handling the problem all at once was a better strategy than a divide and conquer strategy that had been used before, in which you first generate in the lab, you generate long pieces of the genome. You sequence those pieces, but much longer than a read, hundreds of thousands of nucleotide or tens of thousands. Sequence those pieces and then put the pieces together. And then it turned out that instead of doing it in 2 stages with different technologies for the 2 stages, it was better to do it all at once, going directly from the short reads to the full genome. There's also an important variation of this problem that arises because your genome and mine differ in only a tiny percentage of all of the nucleotides. And therefore, if we have a reference genome, then it's much easier to sequence another genome which differs only slightly from it. And so most of the sequencing that goes on in the companies, for example, that are providing genetic information to individuals, most of the sequencing that goes on comes from aligning a new genome, the reads from a new genome against a reference genome, which is an easier problem than doing it ab initio. So in the remainder of the talk, I want to talk about not so much the individual molecules, the individual reads and so on, not so much about sequencing a genome, but trying to understand how the biomolecules that do the work of the cell actually work together, the systems that they form to regulate what goes on in living cells. Here's a quote from a well-known biologist named Gary Odell at the University of Washington. He says, "We can approach understanding how the whole genome works by breaking it down into groups of genes." I would say proteins, actually. It's really the proteins that do the work. Breaking it down into groups of genes that interact strongly with each other. Once researchers identify and understand these network modules, the next step will be to figure out the interactions within networks of networks, and so on until we eventually understand how the whole genome works many years from now. Networks are the fundamental units of natural selection. They are discrete and identifiable objects that are passed down from one generation to the next. So the rest of this talk will be introducing some of the issues that come up in the unfinished work of trying to identify these molecular machines or pathways or regulatory modules that that interact to perform the work of living cells. So what we mean by gene expression is basically the question of which parts of a genome get transcribed into messenger RNA, in what abundance are those messenger RNAs produced, which of them represent genes, in what abundance those genes are produced, and then how the genes work together, or really the proteins work together to perform the work of the cell. So this view, which is the classical one, emphasizes the proteins as the molecules that do the work, but in fact, in recent years, it's been found out that RNA molecules also have significance in these regulatory processes. So there are several levels of regulation. First of all, getting the genome, which spells the names of the proteins in the form of the genes, the parts of the DNA that code for proteins. The transcription of the genes to messenger RNA, and usually the determination of whether a particular gene is being transcribed and in what abundance is regulated by existing proteins in the cell, which have to bind near the gene to enable it to configure itself to be copied into RNA. And then the whole business that we are just beginning to pick apart of the translation of mRNA into proteins and the complex networks of protein-protein and protein-RNA interactions that develop to regulate what goes on in the cell. And then beyond that, we have metabolic processes. The proteins serve as catalysts to regulate complex chemical reactions. And out of all of this collection of chemical processes in the body determines your actual physical traits, your phenotypes, your tendency towards disease, which are regulated by the interaction of many of these chemical processes. So it's a very big, complicated problem. Solving it would entail understanding, for example, the different ways in which different kinds of cancer develop and how we might intervene in attacking them. And it'll be the work of well over a century to understand all of this structure. So I'm going to talk in the remaining time about two key research areas, the analysis of protein-DNA interactions and the analysis of protein-protein interactions. But first, here's just a little example. P53 is a particular protein which is critical in the development of an abnormality in that particular protein is indicates a tendency towards cancer. And here is a little causal network of the proteins related to P53 in a cell, where the boxes and ellipses and ovals represent proteins and the arrows represent some causal influence, which may be either to stimulate a protein or to turn it off. So ultimately, we would like to be able to describe these networks in different types of cells, normal cells versus disease cells, for example. But only in a few cases has this been accomplished at the present time. So I want to talk a little bit about the process of transcription, the copying of genes into messenger RNA. And it's regulated by proteins called transcription factors that bind to DNA, bind to the genome near the start site of transcription and influence the rate of transcription. And the goal is to identify the transcription factors, figure out where they bind to the genome, and determine how they act in combination to enhance or inhibit transcription. This is called the cis regulatory code. Cis is the Latin word for near. It's the opposite of trans, which means across. And cis-regulation means regulatory effects which are on the same strand of the DNA as the gene that's being regulated, on the same side, in other words. Eric Davidson from Caltech is a founding father of the analysis of this kind of transcription using a model organism called the sea urchin, relatively simple organism. And the net effects of his decades of work are to show that the regulatory interactions mandated by circuitry encoded in the genome determine whether each gene is expressed in each cell throughout developmental space and time, and if so, at what amplitude. So in other words, he's succeeded, at least in that organism for some genes, of figuring out in detail how they're controlled. And it's a combinatorial interaction among a number of transcription factors that bind to the DNA. Here's a little cartoon that indicates what we're talking about here. The horizontal line represents the genome, and we see little boxes with the word exon, with the word intron in between. What's going on here is that the gene, which is going to get transcribed, does not consist entirely of consecutive DNA, but consists of sections of consecutive DNA called exons, with parts that don't get copied into the RNA in between, and those are called introns. And we see the little arrow at the first exon indicates the position where transcription starts, but preceding that on the DNA strand are regions, locations, where proteins represented by these ovals, do I have a pointer? Well, I think you can see, bind to the DNA, and they bind at particular locations where characteristic sequences called motifs occur. An important lesson here is that the actual transcript, the actual RNA transcript, is a concatenation of possibly several of these separated exons. And there's a kind of combinatorial logic going on according to which proteins are binding at these recognition sites. So a motif is a short sequence pattern recognized by a transcription factor. These proteins are called transcription factors. And the motifs occur repeatedly in the genome, but with considerable stochastic variation. So the places where a given protein can bind are not determined rigidly by a particular sequence. There are certain parts of the sequence that are strictly necessary, others that are to some extent optional. So you need to work with a kind of stochastic model of the binding sites of these transcription factors. And not only that, but certain combinations of motifs occur, and the transcription of the gene is determined combinatorially according to which combinations of transcription factors are binding. So here we see a little diagram where a particular motif occurs in each of a number of DNA molecules, sometimes on one strand indicated by the left to right direction, sometimes on the opposite strand indicated by the right to left interaction. If we look at all of these occurrences of motifs, we can sort of describe probabilistically what the motif looks like. That's that big diagram in the lower right. And each column represents a particular nucleotide position. And there are certain columns where you have huge letters, the G and the A being the first of those. And that means that the binding site to attract a particular transcription factor has to have G and A in those positions. But in other positions, you can have, for example, in the first position, the leftmost one, you can have either an A or a G in roughly equal proportions. And the heights of the letters sort of represent, in general, the frequency with which a particular letter occurs in a particular part of the motif. So you have here another one of these very hard combinatorial problems of looking at a bunch of sequences where you suspect that a particular transcription factor binds, and look for sequences that occur with some stochastic variation much more frequently than chance in those particular sequences. So there we have a combinatorial problem. I think in the interest of time, well, maybe I'll just say a little bit. The simplest description of a motif within locations would be what's called a position weight matrix. This would be a matrix with a column for each position in the motif and a row for each nucleotide. And in the column, there would be 4 non-negative numbers summing to 1, which represent the frequencies of A, C, T, and G in that particular position. And in the simplest model, these positions are assumed to be statistically independent. So in one version of the motif finding problem, you take the problem to be the discovery of the position weight matrix, from which you can identify the binding sites on several different, of the same transcription factor on several different sequences. So it turns out that there are much more complicated patterns in these sequences which have to be modeled in a more complicated way. Some of these motifs turn out to be palindromic, that they are similar whether read from right to left or from left to right. There are other known dependencies among the columns. And so more elaborate models have been carried out, have been developed by a number of people, including joint work I did with my former student Eric Xing, looking at alternative models. So the idea is that a given protein has a 3-dimensional structure that might indicate the way in which it would be likely to bind to DNA, and that can be taken into account in these models. And a reasonably general stochastic model for a family of motifs is given by what's called a hidden Markov model, a Markov chain in which each state dictates the character of the nucleotide distribution at a given site. So a hidden Markov model is basically a stochastic finite state machine where the states represent the different columns in the motif. You have transition probabilities, and from each state there's a certain probability of generating a symbol. So a hidden Markov model is a Markov chain model for the generation of a family of sequences, and it's a fairly flexible model for modeling these sequences. And there are well-known algorithms. There's an algorithm called the Baum-Welch algorithm, which makes it possible to look at a sampling of sequences generated by one of these Markov chains with outputs. Given the state diagram, you can infer the transition probabilities and output probabilities and get a complete model of a family of sequences.
What are the percentages inside the boxes?
I think those are...
There's some kind of defect from it being amount in equals amount out, but what happens to that stuff?
Let me look.
Inside the diamond?
Yeah.
Like inside C, there's a 0.6, which is presumably the difference between both those equal.
Right.
That get tossed to the trash? Or
Yeah. I have to admit I can't answer that question right off, so we'll let it go. And then beyond that, once we've figured out the individual motifs, there's the whole combinatorial logic of which combinations of transcription factors have to be present. And here is how Davidson finally describes the goal of this kind of analysis. He says, portions of the endo-16 cis-regulatory system of the sea urchin are to date the most extensively explored of any with respect to the functional meaning of each interaction that takes place within them. And here's the interesting part. What emerges is almost astounding: a network of logic interactions programmed into the DNA sequence that amounts essentially to a hardwired biological computational device. So he's saying that with enough experimentation and analysis of these models, you can eventually derive something like a Fortran program or a logic circuit, which tells you, according to which proteins are coming in, the extent to which the transcript is being manufactured. Not everybody agrees that it's so simple, but at least in some cases, his work illustrates that that kind of fairly complete analysis can be done. The other particular application that I want to talk about has to do with protein-protein interactions. So we have already said that the machinery that carries out and regulates the functioning of cells consists largely of sets of interacting proteins. These include protein complexes or molecular machines, and signaling pathways, which are more like straight-line programs where you have a chain of interactions between successive proteins that eventually transform extracellular signals into the activation of transcription factors. So basically, these are networks and graphs indicating interactions between proteins, and we would like to, similar to the example I gave of the P53 protein earlier, and a kind of gold standard problem is to actually to infer what these networks of interacting proteins look like. And of course, they may be changing, they may be stochastic, but they may depend on the particular cells that you're looking at. So the answer may be complicated. Whoops. Oh, what did I do? All right. So here I'm just putting in a little plug for the importance of proteins. They have structural or mechanical functions. They participate in cell signaling, immune responses, the cell cycle, the replication and death of cells, et cetera. And the kind of structures that we're trying to analyze here are what we call protein modules. That is to say, interacting sets of proteins that perform some cellular function. And so a kind of data that's been gathered for some organisms is what is called the protein-protein interaction network of the organism. This is a network with perhaps 10,000 nodes and 30,000 edges, undirected edges usually, in which the nodes are the proteins in the organism, and an edge is present between two proteins if there is evidence of physical interaction between the proteins. So these networks have been codified and stored in databases for a few species, fruit fly, human, a few others. And right now, they're very noisy. There are many false positives, many false negatives. And so the story I'm about to tell is a little bit like a fairy tale. I'm really describing what we could do if these networks were perfect, if we really knew reliably the protein-protein interactions that take place. And in fact, we're hindered at the present time by the amount of noise in these networks. So a protein module is defined as a set of proteins that aggregate to perform a cellular function. Now, if we think of a protein module as a subgraph of the protein-protein interaction network, so the picture you should have now is that there's this vast hairy ball of nodes and arcs where if I tried to draw it, it wouldn't look like anything recognizable. It would look like just a skein of messed up wool or something like that. But it would be essentially a graph, a huge graph with proteins as vertices and edges, protein interactions as edges. And a protein module would be a subgraph of that graph, the subgraph induced by a particular set of proteins. So what would we expect to be the characteristics of one of these protein modules? Well, first of all, the proteins should have a common function. So if you look in a database of functional annotations of programs, of proteins, you would expect to find some consistency in the annotation of those proteins. Secondly, you would expect the network to be connected, to induce you would expect the module to induce a connected graph, because if the proteins are not interacting well enough to be able to reach any one from any other one, they're not likely to form a molecular machine. And it turns out that an additional property is that a protein module should be modular. That's where we get the name module, where being modular means that there's a high ratio between the number of interactions within the set of proteins as compared to the interactions to the outside. So, in other words, they should be tightly interacting among themselves, but not too greatly influenced by what's going on outside. There's a sort of principle of separation. And so we should expect to have the ratio between the number of edges with one endpoint in the subgraph and the number of edges between vertices in the subgraph. We would expect that ratio of external edges to internal edges to be small. And that ratio is called the conductance of the subgraph. Now also, particularly because we have so much noise in any individual protein interaction network, we have the principle that when nature solves a particular problem in evolution, that it tends to reuse that solution. So in the course of evolution, you would expect that, at least for closely related species, a module in one species should have a counterpart in the other.
Roughly how many vertices are in these modules?
Up to 20 or 25.
So really
Yeah. So we have another notion that we have to introduce. We'll say that, so now we're looking at two proteins, one in, let's say, human and one in fly. And we'll say that those proteins are homologous if there's evidence that the two proteins have evolved from a common ancestral protein. And we would expect that if you have a protein module in human and a protein module in fly, then there should be, that do the same job, then every protein in the human module should have a homologous protein in the fly and vice versa. Now, unfortunately, we can't look back in time to find out whether that common ancestral protein actually exists, so we have to use indirect evidence, which is mainly determined by sequence similarity. So you would use a tool like BLAST to find homologous proteins in two species, two organisms. And we'll say that protein modules in species A and B form a conserved module pair if every protein in either module is homologous to at least one in the other module. So in other words, so what we're looking for is connected graphs in the two species with low conductance and such that for every protein in the first subgraph, there's a homologous one in the second subgraph, and vice versa. So this is a combinatorial problem that we have to attack. And we have succeeded in a succession of papers in doing this, and even in finding modules that are conserved across three species: yeast, fly, and worm. Although the most recent work we've done involves only two species, human and fly. I'm going to skip the next part, even though it's mathematically interesting. It's basically, it turns out that an algorithm similar to the algorithm that's used in looking at interesting subgraphs of pages on the internet can also be applied to the famous PageRank algorithm that made Google so famous. A version of that, a localized version of that, can be used to find subgraphs, connected subgraphs of low conductance in a given graph by performing a certain kind of random walk, which is analogous to the crawl that people do on the internet. So I'm going to skip that part and just say that there's a way of constructing low conductance connected subgraphs in a given graph. So with my student Lukman Hodgkinson, we've developed an algorithm for constructing conserved protein modules. It's not a perfect algorithm, but it's been pretty useful. So what we first do is to construct connected sets of low conductance in the first species. These are putative modules in the first species. Once we've constructed them, we might look at the annotation of the proteins within them to see if they satisfy the test of having consistent annotation. And this would give us a set of candidate modules in species A, and now we want to find their counterparts in species B. And so the basic problem that this program attacks is the following. Given a module in species A, find a module in species B which contains homologous proteins to the proteins in the first species. And I'm running out of time, so I'm going to talk about a purely combinatorial problem that is abstracted from the goal of passing from module A to its counterpart in the second species. So we formulate it as in the following way. Given a set of proteins in species A, namely the proteins of a particular module, and the protein-protein interaction network of species B, find the subnetwork in species A, in species B of minimum size, which is connected and contains at least one protein homologous to each protein in the first set. So we're just looking for a small subgraph which is connected and which carries over the proteins from one species to the other. And so a purely graph-theoretic version of the problem would be the following. You're given a graph G, you're given a set of colors, finite set of colors, and for each color you're given a set of vertices that can be assigned that color. So the colors are the types of proteins. And the problem then is to find the connected subgraph in the given graph with a minimum number of vertices such that the colors, I've written this in too complicated a way. Let's just say that every color in the set C can be assigned to some vertex in the graph. Every color occurs in the graph. So I wrote this in a too elaborate way. So basically, to restate it, you have a graph, you have a set of colors, you're looking for a connected subgraph. Every vertex is labeled with a color, and then you're looking for a connected subgraph that contains at least one occurrence of each color. And it turns out that there are a number of algorithms that can be used, but they're very costly. So integer programming can be used. That's linear programming with the additional constraint that the variables be integers. I don't have time to explain the integer programming algorithm. You can use dynamic programming, and that'll work if the number of colors is small. But in practice, we prefer to use a simple heuristic which has no theoretical analysis, but works very well. And I'm going to describe it and give you a 1-minute version of that heuristic, and that will be the end of my talk. So here you see a selection from a poem by T.S. Eliot, "Let us go then, you and I, when the evening is spread out against the sky." What does that have to do with biology? Well, it's just a convenient way of constructing examples. So we'll think of this as a grid graph with, I think, 8 rows and 8 columns. So it has 64 nodes. The letters of the alphabet are the colors. And what we're looking for is a connected subgraph that contains, let's say, exactly one occurrence of each color. Okay, so that's the fundamental problem. And it's a very amusing problem. I've spent many hours on airplanes solving instances of this problem to while away the time. I've even thought of making it a cell phone app, but I haven't gotten anybody to do any coding for me so far. So what is the nature of this heuristic? Well, basically, the idea is that it's the less frequently occurring colors that will give the most information about where this subgraph is located. And so what we do is count the frequency of each color. So the red letters correspond to the frequent, the red positions correspond to the frequent colors or letters E, T, N, and A. So what we do is to delete the frequent letters from the graph, and then we're left with connected components. We can delete certain connected components going back, induced by the infrequent letters, the black positions. We find enough connected components to cover all of the infrequent letters. We find a minimal way of putting the frequent letters back, one occurrence of each, and that has allowed us to prune away part of the graph while maintaining the key properties that it's still connected and it still contains at least one occurrence of each letter. And then we repeat this process. We count the frequencies in this subgraph. It turns out that the frequent letters are A, T, N, and I. We delete those. We go through a similar process. And in the end, we finally end up with a connected graph containing exactly one occurrence of each letter. So that's a very quick and hand-waving introduction to the heuristic that we have found to be useful in solving this problem. So I'll stop here, and I'll be happy to take a few questions.
What's your opinion on the incomputability of biology? For example, there's some nascent work by the late Robert Rosen based on category theory that looks at whether the space of Turing machines is flexible enough to encompass biology. I was wondering if you had any opinions on that type of research.
Well, so the question is biology computable? So far, we haven't shown that biology is computable. One of my colleagues goes around giving a talk called Can We Model an Elephant? And he asks if we bring to bear all of the information that's available in the various databases, can we give a complete description of how an elephant works at the physiological and molecular level? Or can we even model a worm? And so far, we don't know how. Certainly, the techniques that I've been talking about here are reductionist. They're sort of local in nature. They say, well, we're going from some very complicated thing called a cell, and we're saying we are maintaining that it can be described in terms of graphs and subgraphs. Well, it's a physical object. It has a spatial dimension. It has different compartments. It has flow in between. How can we model all of this stuff? At some level, we have to model more than just the combinatorics of how the proteins interact. So there's a whole level of dealing with the geometry of the cell, thinking of the forces, the energy that's involved. And which cell are we talking about? They're the same. The cells occur in different parts of the body plan, and they have different roles. So what are the differences between them? They contain different proteins. How can we have a single model that captures all of this? So in addition, and there's something holistic going on that we have to understand. Whether we can actually do this is and get something predictive for a full description of the physiology and molecular biology of an organism is very unclear, very unclear. But if we can get partial answers, if we can find out that certain mutations cause cancer and that we can cancel the effect of those mutations by certain interventions, that's a good thing, even if we don't answer the whole question. So I know that I'm not going to live long enough to get full answers to these questions.
I think we have time for maybe one more quick question. Any other questions? Okay. No further questions. Please remember there's another talk tomorrow in an entirely different building, way on the opposite side of campus. But in the meantime, let's thank Professor Karp again.
Thank you.